Hello friends, I hope you like my videos and it's helpful to enhance your knowledge. If you are having any question, you can ask me in the comment. So in this video, we will discuss about the handling of out of specification results in pharmaceutical industry, right? Friends, my YouTube channel name is Pharma Quality. So please view, like, comment and subscribe my channel to get the knowledge about the pharma quality standards and practices. So now in this slide we will discuss what is out of specification. So a simple definition of out of specification is a test result that falls outside the established specification or acceptance criteria. So now let's discuss with one example. Suppose you are performing an assay test of a drug product and your specification limit is 95.0% to 105.0%, right? And now your results for the assay test is observed 105.1% at higher side or 94.9% at lower side. Then it shall be considered as a out of specification test results and you need to initiate the OS and investigate it why OS results was observed, right? So before going into the deep of OS investigation, we will discuss about some basic definition which are commonly used in investigation. So first is what is assignable cause. Cause which has been scientifically established with conclusive evidence and documented during investigation. That means you have performed the investigation and simulate the results with initial OS result. So assignable cause is a specific identifiable source of variation, right? Next is what is root cause. So cause which is scientifically proven and supported by evidence. So it's not simulated but fundamental reason for a problem is identified. Next is what is probable cause. So cause which is likely to have resulted in or contributed to an OS test result supported by scientific rationale and may or may not be established with evidence. So probable cause is likely reason that contributes OS based on available evidence, right? Next is what is obvious laboratory error. So an observed or instantly identifiable laboratory error that causes the OS, right? So the mistake that made during laboratory procedure or testing that called the obvious laboratory error. Next is what is laboratory error. An error associated with the performance of a test procedure or due to laboratory equipment malfunction or failure. So in the laboratory, there will be some test procedure error or equipment malfunction related error. That is called the laboratory error. Next is what is human error. So error associated when a mistake is made by analyst. So it may be not followed the method of analysis which may include the pipetting error, weighing error or dilution error. So here are the guidance for the industry related to out of specification. So first is investigating out of specification test results for pharmaceutical production. So this guideline is for the US FDA regulation, right? So next is presentation of out of specification investigation by MHRA. So this guideline is for the MHRA regulation. Next is EMA guideline on the investigation of bioequivalence. So this guideline is specifically talks about the bioequivalence studies for the EMA regulation, right? Next is ICH-Q7, that is Good Manufacturing Practices Guide for Active Pharmaceutical Ingredient. So this guideline is not specifically focused on the OS results. Basically, these guidelines provide the principle and guidance for the GMP practices for the API, right? Which include the quality control processes and that may involve the handling of OS results. Now in this slide we'll discuss the details of workflow that is handling of out of specification, right? So first of all, OS results shall be initiated either manually through paper-based procedure or automated systems such as TrackWise or QMS system, right? Next, OS shall be reviewed by the user department head. So user department head can review the details of OS submitted by the initiator, right? And further, this OS will move to the QA review, and QA review shall review the OS record and provide the comments. And after QA comments, lab investigation can be initiated. So QC section in charge or their designee shall perform the laboratory investigation, right? And after the investigation has been completed, so the outcome of laboratory investigation shall be reviewed by the independent investigation team and provide their input. Further, if any root cause is identified, then lab investigation can be concluded, and based on that, further action can be taken, right? In similar way, production section in charge or their designees shall proceed for manufacturing investigation based on the lab investigation outcome and QA input, right? And further, the outcome of manufacturing investigations shall be reviewed by the independent investigation team and provide their input. Then after, if any root cause is identified, then manufacturing investigation can be concluded, and based on that, further action can be taken, right? So based on overall investigation, the required action plans shall be proposed here. And then after that action plan shall be allocated to the respective user. And after the allocation of action plan, the action plan shall be executed, right? So after execution of action plan can be closed and kappa decided can be initiated, and the effectiveness monitoring of that kappa is also initiated, right? And further, the QA cell mention the details of summary and conclusion, and finally QA cell done the final closure. So now we'll discuss the phases of OS investigation. So basically there are two phases. First is phase 1, that is laboratory investigations. So whatever the laboratory related investigation, that will be included into the phase 1. And second one is the phase 2, that is full scale OS investigation. In the phase 2, production and process related investigations shall be done, right? So here we'll discuss the phase 1 laboratory investigation. So first of all, OS results observed by QC analysis. So at the time of result reporting, results can be checked against the specification limit, and if it is out of specification limit, then it shall be considered as OS results. After that report, the US OS results to the section in charge or designee. So after reporting of OS results, analysts shall immediately inform the OS results to their supervisor, and then OS should be logged within 1 working day. So immediately OS should be raised, right? And then all batches impacted by the testing shall be put on hold. So after OS results observed, all batches which has impacted due to the OS results shall be put on hold for further processes, right? And then retain all the test preparation related to OS results such as sample solutions, standard solution, and glassware used for the investigation. So after OS results observed, all analytical preparation, including used pipette, volumetric flask, or any stuff which is used during the analysis, shall be kept aside so that it can be helped during the investigation, right? Next, instrument used will be kept on hold for further uses, right? So also the instrument used for the analysis shall be put on hold to evaluate the system performance as a part of investigation. So now here we will discuss a few examples of what can be considered in a preliminary laboratory investigation for phase 1. Right, so I'm explaining these just for the training purpose, and this example is based on the OS results observed by the using SPLC instrument. So investigation can be different based on the nature of OS observed. So you can see the following action we can be able to take in the laboratory investigation. So first is examine all test article reagents and solution and equipment used. So section head will check all analytical solution, reagent and equipment for any discrepancy, right? Next is check sample handling and storage condition. So handling of sample procedure and its storage condition should also be checked for any discrepancy. Next, check glassware use. So whatever the glassware, that is volumetric flask, pipettes, or any glassware that has been used in the analytical preparation, that should also be checked. Next, check the instrument calibration status. So the used instrument, that instrument is within calibration due date or not, that should also be checked. Next is system suitability. So check any discrepancy in the system suitability solution if it is applicable, right? Then check the solution preparation records as per the testing procedure. Next is check the reference and working standard uses distinguishes. So whatever the standard analyst has used in the analytical preparation, that should also be checked. Check the solvent and reagent details. So whatever the solvent and reagents has been used in the analytical preparation, that also be checked. Next, check the chromatograms and spectra of that particular injection. Check the calculation of the results, that is all the calculation has been done correctly and outcome of that calculation is accurate. Next, check qualification status of analyst. So analyst who has performed the analysis and OS result is observed, that analyst is well qualified or not. So the objective of preliminary laboratory investigation is to identify obvious error or probable laboratory errors. So if obvious error is identified as transcription error or calculation error or documentation error, then the result shall be corrected and documented, and that result shall also be reviewed by the QA, right? So various hypotheses we can plan to rule out the laboratory error, right? So to check the instrument malfunction, re-injection of the retained sample and standard preparation shall be planned. So after re-injecting, if the same OS results are observed, then you can conclude that there is no instrument malfunction. That is, instrument reproduce the same OS results, right? Next is to rule out the vial filling error. So refill the HPLC vial from the original and final sample solution and inject the solution, right? If the same OS result are observed, then you can conclude that there is no filling error. That is, analyst fill the vial correctly, right? Next is to rule out the inadequate mixing of prepared solution. So for this, resonicate the solution or manual shake the solution based on the testing procedure and inject the solution, right? If the same OS results are observed, then you can conclude that there is no uh, you know inadequate mixing of solution, that is, analyst mix the solution correctly, right? Next is to rule out the dilution error. So redilution shall be done from the stock solution and inject the solution. So again, if the same OS results are observed, then you can conclude that there is no dilution error, that is, analyst perform the dilution correctly. So these are the few hypotheses we can plan to rule out the laboratory error. So whenever multiple samples are tested together, the impact on all sample must be assessed, right? So if analysis is performed for more than one batch, then impact on all those samples should also be done. If laboratory error is confirmed in hypothesis testing, then retesting shall be carried out, right? So if any laboratory error is identified, then retesting of OS batch and all impacted batches shall also be performed. If in these above preliminary investigation no cause of OS is identified, then extended investigation shall be carried out, right? So if no cause is identified during preliminary investigation, then further investigation shall be carried out to identify the root cause. Along with this, phase 2 full scale OS investigation shall be carried out. So also phase 2 investigation shall also be started, right? So now we will discuss the phase 2, that is full scale OS investigation. So phase 2 of OS investigation is conducted if phase 1 does not identify any assignable cause for the OS results, right? So generally phase 2 focuses on investigation that extends beyond the laboratory, that includes a thorough review of the manufacturing process. So let's discuss what is phase 2 full scale OS investigation and what we can include into this investigation, right? So first of all, you can see review of batch production records. So first, batch manufacturing records we can check. So check the batch records for any kind of deviation or discrepancies observed during the production processes. Next, we can review the critical process parameter. So all critical process parameter were within the their specified range throughout the production or not. So that things we can be checked, right? Next is in-process controls. So review the in-process control results to identify any trends or issue that might have contributed to the OS results. So in the batch production records, such things we can check. Next is material and equipment evaluation. So first one is the raw material. So we can assess the quality and source of raw material used in the batch. So we can check also for any changes in the supplier or specification or any batch numbers, right? Next is equipment logs. So ensure that all equipment used in the production and testing processes was properly calibrated and maintained, right? Next is cleaning records. So review the cleaning and maintenance record of the equipment to rule out the contamination related issues, right? So next is personal and training record, right? So here you can check the operator logs and operator interviews. So in operator logs, check the training records and qualification of personnel involved into the production and testing process, right? Next is conduct interviews with the operators and technician to gather additional insight and observation that may have been overlooked. So you can perform the interviews of those operator who has involved into the all production processes, right? Next is environmental condition. So facility condition we can check. So review the environmental monitoring records for the production and storage areas. So we can focus on factors like temperature, humidity and cleanliness. So these things we can verify and investigate any environmental excursion that may have occurred during the production and storage of the batch. So these all related to the environmental condition. Next is comprehensive testing review. So retesting and resampling. So if appropriate, perform retesting or resampling according to the established protocol to confirm or to refute the initial OS results, right? Next, method validation. So ensure that analytical method used are validated and suitable for their intended purpose, right? Next is reagent and solutions check. So verify that the quality and expiry date of reagent and solution that is used during the testing, right? Next is historical data and trend analysis. So compare the current OS results with historical data of previous batches to identify any reoccurring issues or trends. Next is control chart and trends. So use the control chart and trends to visualize the process stability and detect any patterns that might explain the OS result. So these all things you can include into the phase 2 OS investigation. So now in this slide, we will discuss about the practical approach to make an investigation report for OS results, right? So here I will take a simple example, that is OS observed in assay test, and try to explain it, right? And I will explain this just for the training purpose. there may be the different investigation scenario based on the nature of OS, right? So first one is the event description. So here you need to elaborate the problem statement, that is details of OS observed. For example, if you have observed the OS results in SS test, so for this OS you need to describe the details, right? And also you need to give the details such as OS number. So OS number can be assigned based on the internal process, right? And also provide the details of product name or material name, batch number, AR number, market of product, stage, manufacturing and expiry date of product or material, date of analysis, date of OS occurrence, and date of initiation. And apart from this, also need to mention about the specification number and on which test the OS results was observed, and result observed details against the specification limit. And if there is any SOP number is also applicable, so you can give the details for this SOP, right? And apart from this, who has identified that OS and how and where it was identified. So such things we need to elaborate into the event description, right? Next is immediate action. So what action you have taken immediately, that things we need to elaborate here, right? So here you need to describe about the action you have taken immediately after identification of OS. For example, OS results is informed to the section head or department head. So that action you have taken, so you can elaborate here. And also you can elaborate the that instrument you have used and analytical preparation that was hold for the further investigation, right? So such things we can elaborate here. So next is initial impact assessment. So here you need to elaborate about the initial impact with respect to the product quality and impact on batches, right? If the product is already commercial, then FAR, that is field alert report, generated or not. So such things we should also evaluate. And impact on other batches should also be evaluated if other batches was analyzed along with this OS results, right? Next is investigation teams. So here you need to define about the person who are involved into the investigation, such as name of person, their department and their designation, right? So the next is investigation finding. So in this, first is process flow. So here you need to define about the process step details and what can be the possible error for the OS results, right? Next is historical check. So here you need to provide the details of similar type of OS with root cause and kappa, right? So you can consider the last 2 year data to check the same type of OS and provide the details, right? And also we need to elaborate the batch history record for any OS or discrepancy observed in the API or excipients used during the product manufacturing for which OS result is observed, right? Next is data collection and interpretation. So here the data can be collected and interpreted based on the OS results observed, right? So here are a few examples we can discuss. So first is analyst details. So here analyst training and qualification status can be checked to evaluate that analyst is competent in performing the respective test or not, right? Next is analytical data review. So here test data sheet can be checked to verify that analyst has prepared and performed the analysis as per the testing procedure, right? Next is physical verification of working bench and instrument. So here we can check to verify that physically that glassware used in the preparation that is correct or not, labels pasted on the glasswares that is correct or not, and the instrument conditions is okay or not. So such kind of things we can check here. Also with respect to the instrument, is there any leakage or any kind of air bubbles trap into the port, So such kind of things we can check, right? Next is column review. So correct column is used or not as per the testing procedure, and its dimension is okay or not. So such thing we can check. Next is review of method of analysis. So testing method as per the testing procedure are correct or not, such as instrument program, mobile phase preparation, diluent preparation, and standard or sample preparation, and other things as per the testing procedure are accurate or not, we can check into this, right? Next is review of chemical and reagents used. So chemical reagents used, that was within the validity or not, that we can check. Next is standard verification. So working standard or reference standard used, that were within the validity or not, and other details such as, you know standard batch number, validity period, and make shall also be evaluated. So next is sequence review. So here we can check any transcription error related to sample or standard weight or batch number or AR number, etc. Right. Next is pressure graph and chromatogram review. So here we can check the pressure channel for any fluctuation in the pressure or not, right? And check the chromatogram for the any retention time shifting is there or not, right? Next is area response review. So here we can check any area variation as per the previous trend, right? So based on these above preliminary investigation, if any laboratory error or cause is identified, then we can conclude the OS, or further we can plan the extended investigation that shall be carried out, right? Now the next is root cause assessment. So further hypothesis testing can be performed to identify the root cause if there is no any assignable cause is identified during the preliminary investigation, right? For example, hypothesis is planned for the same vial, refilled vial, and redilution of OS solution, and result for same vial and refilled vial are found in line with the initial OS results. So then instrument error and vial filling error shall be ruled out. However, let's say if the redilution of oil results observed within the specification limit, then we can say this dilution error is contributed to the original OS results, and assignable cause is related to the dilution error only, that is analyst error, right? Further, various investigation tools can also be used to identify the root cause of OS, such as fishbone diagram or 5Y tools, right? Next, once you concluded that this is the analyst error or human error, so human error evaluation shall be done. If there is no any human error or analyst error is applicable, so human error evaluation section shall not be applicable, right? So if root cause is identified as human error, then name of person, their employee code, and what error he or she has done. So such details we need to mention, and we can use here also the 5Y tools, that why analyst has done the dilution error. So in such a way we can investigate. Next is retesting. So based on the identification of assignable cause, retesting of OS batch and also the other impacted batch shall, the testing shall be performed, right? Next is root cause conclusion. So here, based on the detailed investigation, you need to elaborate the root cause detail. So assignable cause for initial OS results is due to the analyst error, and that is analyst done the dilution error. So such thing we can define, right? Now the next section is impact assessment based on the root cause identified and batch disposition, right? So here you need to elaborate about the details of impact with respect to the product or batches, right? Since this OS is observed due to the analyst error, hence there will not be any impact on the batches. So in such way we can elaborate. Next is kappa and effectiveness monitoring. So here you need to describe about the what corrective action and preventive action based on the identified root cause, right? And also you need to open an effectiveness monitoring of the kappa to check the kappa is effective or not in the future, right? So in this way we can perform the investigation. So friends, this is all about the out of specification. So in this video I have explained the concept for the training purpose, right? So basically OS investigation shall be carried out based on the what kind of OS is observed, that is for which test and by what type of instrument used for the testing. So I hope you can understand the concept of OS handling. Thank you for the watching video.